Hey, welcome to the Biochemistry Academy. In today's video, we'll talk about enzyme kinetics. There are many people who have been looking for videos like this. Well, we will start from the scratch to the advanced topic on enzyme kinetic. So first, we would understand what enzymes are. And we say that enzymes are simply biological catalysts that speed up biochemical reaction. And the way they do this is that they lower the activation energy of the reaction and they are not also consumed in the reaction. So this is how an enzyme lower the activation energy of a reaction. So this is a graphical representation in the presence of an enzyme and in the absence of an enzyme. So let us proceed. So this is an enzyme E, and this enzyme acts on a molecule, and this molecule is called a substrate, and we represent it as S. So to form an intermediate, this intermediate ES, also called enzyme substrate complex, and when it forms this intermediate, then the substrate is converted into a product P, and the free enzyme go back to the original form, because we said that enzymes are not consumed in the reaction. So to understand this more, we talked about enzymes in our playlist on enzymes. So to have a better understanding or a full understanding of enzyme, the types, the classification, coenzyme, cofactors, you can simply go back to our playlist on enzyme and watch it, or you can just quickly skip this if you already have knowledge of what enzymes are. So there was a thought and a need to understand the speed at which an enzyme works. That is where enzyme kinetics comes in. Enzyme kinetics is the study of how enzyme binds substrate and convert it into a product. So imagine you have 10 workers, and these 10 workers are the enzyme, and you give them a small pile of work. Now this small pile of work represents the substrate, and then there was a need for you to measure how fast they were able to finish it. So it's like the same thing. So measuring how fast they finish it is just like measuring the velocity of how an enzyme works. So enzyme kinetics involves doing the same test, measuring the velocity at many different substrate concentration. The foundation to understand enzyme kinetic is that enzyme concentration is constant, but the substrate concentration is always changing. Now, the study of enzyme kinetic becomes easy when we try to understand the reaction by plotting a graph. So we are going to use the reaction E plus S, which gives us our enzyme substrate complex, and then it is converted to the free enzyme and the product. So in respect to time, we can plot this graph as the concentration of substrate or product per time. So let us look at the concentration of the substrate first. Now, as the substrate is consumed, the concentration of the substrate decreases over time. And as product formation begins, the product concentration increases over time. So it is more easier to understand this by plotting a graph. So if we take the slope of the graph, we have the rate of the reaction. Now, The rate of the reaction will be the change in the substrate over time and the change in the product over time. We see that as the substrate concentration is reducing or decreasing, we have a negative sign which shows the decrease in the substrate concentration, while in the case of the product, it is a positive sign because the product's formation increases over time. Now, the unit of the rate of reaction is concentration over time. It is also called the velocity of the reaction, as the concentration is changing in respect to time. If you remember during our elementary time in school, like in our secondary school or in our high school, whenever we see distance over time, we are talking about velocity. So bringing it to biochemistry here, when there is a change in either the substrate concentration or the product concentration over time, it is also talking about the velocity of the reaction. So let's look at the graph. As the velocity of the reaction changes when the substrate concentration change, when there is an increase in the substrate concentration, the velocity of the reaction also increases until it reaches a plateau. And when it reaches the plateau, it is called maximum velocity or Vmax for the particular enzyme. So to find the substrate concentration, we will divide Vmax by half. Now, the substrate concentration at which half of Vmax is achieved is called Km. Normally, students make mistake when they ask them to define Km. So we say Km is the substrate concentration at which half of Vmax is achieved. So here comes an important term called Kcat. So Kcat is the number of substrate molecules converted into products per active site per seconds. It is also called turnover number. The unit is seconds inverse. So the ratio of Kcat over Km represents the efficiency of the enzyme. The ratio is called specificity constant, meaning that at high ratio, the enzyme will be very efficient. In our next video, we will continue the course on enzyme kinetics and we will talk about the Michaelis-Menten equation and the derivation of the equation. Please don't forget to like, share, and subscribe to the Biochemistry Academy for more content like this. Arigato.